Welcome back. This is the second presentation on communication, integration, and homeostasis. In this lecture, we will talk about some of the more novel chemical messengers and systems, and then begin a discussion of homeostasis control mechanisms. So a few of the novel signal molecules will be discussed here briefly. Calcium was mentioned in the previous presentation on this topic, and let's look at it in a bit more detail here. So we begin first, of course, calcium is at a certain concentration in the extracellular fluid. It is regulated in the interstitial fluid and the plasma by hormonal systems that we ultimately will discuss. So it's not just one calcium molecule. I always want to draw your attention to the fact that there's a concentration there, and very low concentration of free calcium in the cytosol. So that's a very important thing to remember. So calcium is very dangerous when it is just floating around unbound or not contained in a membrane system like the ER inside a cell. So there are many mechanisms to keep it from being floating around in the cytosol. But what this creates is a very high concentration gradient. Regardless of how little calcium is outside the cell, there's even less inside the cell in the free form floating in the cytoplasm, the dissolved calcium ion. So keep that in mind. Okay, so let's move on then. So we have that certain extracellular calcium concentration, a concentration gradient across the membrane. If the channel, the calcium specific channel, ion channel, is open, then calcium is going to move into the cell. So these might be a voltage gated calcium channel, for example, as in an axon terminal. Excuse me. And so we also can have, from an electrical signal or other mechanisms, calcium released from the intracellular stores. So we saw that through some of the chemical signaling mechanisms, just globally reflected here with this symbolization below. So two sources of calcium, intracellular or extracellular, and the target is to become free calcium, the divalent cation, dissolved in the fluid portion of the cytoplasm, the cytosol. So the cytosolic free calcium concentration is a signal. So when this increases, and it's nearly zero, it's very tightly regulated to be at low levels. So these are transient signaling events inside the cell. So you'll have a transient calcium concentration increase in the cytosol. And the reason it's transient is because something's going to be there to have the calcium bind to it. So these calcium binding proteins then perceive that. Anyway, proteins don't really perceive things. But anyway, have the capability of binding that calcium and responding in a way such that it can lead to other events. One example of a calcium binding protein is calmodulin, and there are other types as well, calbindin and calpains and things like that. So this alters the protein activity. So think of it this way. It's in the unligated form, and then when the calcium is present in the cytoplasm, it becomes ligated. It's not a covalent bond. The calcium is not stuck to it forever. It can come off. And the calcium coming back off these molecules is, at least in part, going to be dependent upon the free cytosolic calcium concentration, or cytosolic free calcium concentration, get the sequence of those words correct here. And that was going to be regulated by other mechanisms. Remember I talked about that ATP-dependent calcium pump here that's going to pull calcium, the free calcium, out of the cytosol and back into the intracellular stores. There may even be pumps to pump it out as well. So as soon as you get calcium going into the cytoplasm, it's going to bind to things or be removed, and then you have a low calcium concentration again, and so that calcium can come back off. So these are typically transient events, depending upon what changes in these proteins. So it may change the conformation of this protein so that it ends up, really grabbing that calcium and holding on to it for quite a long period of time. So there are modifications on the general scheme that I gave. But once the calmodulin binds calcium and becomes activated, it doesn't stay active forever. There's no sense in a signal if it never ends. So there are other calcium-binding proteins, as I said, as well, that might be associated with exocytosis or movement, such as the calcium-binding proteins of the filaments within a muscle. Some of these novel signal molecules are gases, like nitric oxide. So the nitric oxide activates guanylyl cyclase and leads to the formation of cyclic GMP, and it acts as a neurotransmitter or neuromodulator in the brain. It also has tremendous vascular effects produced by the endothelial cells, and it diffuses to the nearby smooth muscles in the wall of the blood vessels and causes vasodilation. So it had been known for a long time that there was an endothelial dependent vasodilator signal in blood vessels, and for a couple of decades at least it was not identified. The reason was nobody expected it to be a gas. It turned out to be nitric oxide. So there are enzymes that will generate nitric oxide, actually from an amino acid, arginine, in the endothelial cells, and when triggered, when those enzymes are activated, they'll take the arginine as a substrate and end up producing nitric oxide. Now the nitric oxide is going to activate guanylyl cyclase and increase cyclic GMP within the smooth muscle cells, and then you get a relaxation of the smooth muscle cells and a vasodilation. So I know I went into detail here, but I just wanted to fill in some of the blanks. Carbon monoxide is also a chemical messenger, activates guanylyl cyclase and cyclic GMP, targets smooth muscle and neural tissue. Hydrogen sulfide even targets cardiovascular system, and garlic is a major source of precursors for the production of hydrogen sulfide. That's why garlic smells so badly, right? So the lipids are another example. So under the class of lipids, arachidonic acid is one of the chemical messengers. There are others, and there is a cascade for the production of these. So metabolic pathways that involve enzymes that convert substrates to products. So we have a membrane phospholipid that's acted on by PLA2. So this is different than the PLC that we saw before. Phospholipase A2, the PL is the phospholipase. So phospholipase, you ACE it, you act on it as an enzyme and generate another molecule, arachidonic acid, that itself can be a second messenger, can affect second messenger activity, and it also can be acted on by cyclooxygenases like COX-1 and COX-2, cyclooxygenase 1 and 2, two different genes for two different gene products, isozymes, right? Different forms of the same enzyme, isozyme. And these will generate prostaglandins and thromboxanes. And then lipoxygenases will form leukotrienes, and then these would be lipid-soluble chemical messengers acting predominantly in paracrine fashion. Again, there's variations on that scheme of the distance, if you want to think of it that way, over which these can diffuse, remain active, and produce responses. But typically they're going to be localized inflammatory events. So the leukotrienes play a role in asthma and anaphylaxis. So this is what we were talking about, the parameters associated with the distance over which these molecules can get away from their source and remain active, not be inactivated by mechanisms, typically degradative enzymes, and reach target cells over a diffuse distribution. Or there's the other possibility that many cells are giving rise to these, so you get a global effect that's supposed to be localized, but cells all over the place respond. So two ways of thinking about that. A pocket of cells that can produce enough to affect many cells, or many cells responding when it's only supposed to be a pocket of cells that respond. I hope you get that distinction. Because we have these in our body to help us respond normally to environmental challenges, and when the response is inappropriate, then you have asthma and anaphylaxis. Leukotrienes do not exist simply to generate asthma and anaphylaxis. They contribute to normal inflammatory responses that protect us on an ongoing basis from certain types of environmental challenges. So the prostanoids are other sets of lipids, including the prostaglandins that are involved in sleep, inflammation, pain, and fever. Thromboxanes are others here. Non-steroidal anti-inflammatory drugs will target some of these cyclooxygenase molecules. They are typically inhibitors. So the NSAIDs, non-steroidal anti-inflammatory drugs, typically are COX or cyclooxygenase inhibitors. Some can inhibit only COX-1 or only COX-2, or some are more general and they inhibit both of them, and they help to prevent inflammation. So acetylsalicylic acid and aspirin, ibuprofen and Motrin are a couple of examples. What's the active drug compound in Tylenol? It's escaping me right now. It differs because it is an analgesic, but it doesn't inhibit the cyclooxygenase enzymes. Celebrex is another one you might have heard about. Anyway, there are some specialized drugs that are not available over the counter. They're available only by prescription that also inhibit the cyclooxygenases and help to act as anti-inflammatory drugs. Let's then begin to think about regulatory mechanisms in the context of homeostasis, and the first thing we think about is modulation. So I've been throwing this term around, and let's think about it a little more directly. So we need to think about specificity and competition, agonist acting agents versus those that act in the opposite direction as antagonists, multiple receptors for one ligand, so more than one gene to produce a set of proteins that can be specific for a particular agonist or ligand, but have different effects, such as the alpha receptor for the adrenergic molecules, such as norepinephrine and epinephrine, the accepted terms for those substances. We no longer use the terms adrenaline and noradrenaline. We use epinephrine and norepinephrine, respectively. I don't know why they changed it that way. I like the adrenaline, noradrenaline better. And then you have alpha adrenergic receptors, so I don't know who names these things, but that's the way it goes. So the alpha receptor typically is associated with vasoconstriction, and the beta receptor, the opposite, vasodilation. The agonist versus antagonist concept are basic ideas shown on this slide. There are variations on this, so don't get locked into these basic concepts as the strict or only way that you can think about drugs that bind to receptors. And also think of exogenous, coming from outside of the organism, synthetic molecules, or even of other biological origins, like the opiates, that that act on the receptors for the endogenous molecule, the opioids, for example. So there's variations on this. This is the extremely reduced, simplified form. So the primary ligand acts on a receptor. So let's say it is an opioid acting on an opioid receptor. Well, an agonist will activate that same receptor. So it might be morphine or heroin acting on the same opioid receptor, but it is an opiate, O-P-I-A-T-E, or opiate, however you want to pronounce it. I exaggerate to help put it in your mind that there are different things, so don't always pronounce it the way that I do. Okay. I'm just trying to help the learning process. I hope it works. So an antagonist blocks the receptor activity. So now this is where I will take the moment to give a more extensive definition on this. There can be different ways that antagonists work. One term for some of them is called a silent antagonist, where it can be present and it can even bind and unbind or associate and dissociate from the receptor, but it doesn't prevent the endogenous ligand from having its basal effect. So therefore you can be taking this medication and it's silent unless something happens to where there's suddenly more of the ligand or agonist, endogenous agonist, present, then it will block any additional activation of the receptor. And then there are strict antagonists that come in and they block this. It doesn't matter how much. I mean, it's always a concentration dependent relationship. But anyway, you can think of it as a strict antagonist. Then when it's there at a sufficient concentration, prevents any endogenous ligand from binding. So it will inhibit even the baseline stimulation of the receptors. So those are kind of important distinctions, even that one example. Clinically, it's also very important to understand, and the opiates are a good example. If somebody overdoses on heroin and they can get to the emergency room quickly enough, there are antagonists that can be given, the strict antagonists that can be given to the individuals that will block any effects of exogenous, the heroin, and the endogenous opiates. And they know, they have a biological activity over a known period of time, and they know what doses to give and stuff like that. So someone can be basically unconscious because of a heroin overdose, and give the antagonist, and they come out of it, because the strict antagonist removes all activation of the enzymes by the ligands. In that case, it would be an agonist. The heroin would be an agonist to the endogenous opioid receptor. So hopefully those examples helped you understand that. It's a great system to think about, the opioid-opiate system, to make that point. So continuing on with this idea of the modulation of signal pathways, let's think about the effects for a target response that depends on the target receptor. So the alpha and the beta receptors, the adrenergic receptors, are great examples. So you see here they're using the term epinephrine rather than adrenaline, but that is an alpha adrenergic receptor and a beta 2 adrenergic receptor that they're talking about. So there's actually 4 genes for 4 different proteins, an alpha 1 and an alpha 2 adrenergic receptor, and a beta 1 and a beta 2. In this slide, we're only thinking generically of an alpha receptor, the alpha 1 receptor actually, and the beta 2 receptor. So they're expressed on the smooth muscle, the skeletal muscle of a blood vessel. Okay. You heard me. I just said smooth muscle lining the wall of, or within the wall of a blood vessel. And then it said skeletal muscle. But this is a blood vessel within a skeletal muscle. Okay, and this is a blood vessel within the intestine. So we have an intestinal blood vessel containing an alpha receptor and a skeletal muscle blood vessel containing a beta 2 receptor. It is the smooth muscle in the wall cells in the walls of these blood vessels that are expressing these receptors. Okay, so differential gene expression from the same type of cell in different organs, but within the same tissue. Right? So hopefully you recall the different forms of gene expression. There's cell-specific of the whole set of developmental and things like that. But we always want to think of cell-specific gene expression, tissue-specific gene expression, and then sometimes organ-specific gene expression. And here we're seeing cell-specific gene expression, the smooth muscle cells, but of expressing the adrenergic receptor genes, whereas the connective tissue cells are not expressing this receptor. So we have a cell difference based upon the adrenergic receptor. But it is tissue specific to the blood vessel as well. So the smooth muscle cells in the tissue, the blood vessel lining or the wall, but we have organ specific gene expression in the smooth muscle cells of the tissue, the vascular tissue of the organ. The skeletal muscle expresses the beta 2 receptor gene, and that in the intestinal wall, the alpha. So that should have brought in a lot of the other information, but I need you to think about these things because this is the way that I teach physiology. You need to understand it this way rather than this simplified illustration. Anyway, so the same molecule comes out of the adrenal medulla, right? The epinephrine. So the epinephrine is in the plasma, diffuses out, binds to the alpha receptor on these intestinal blood vessel smooth muscle cells, and it causes vasoconstriction. In skeletal muscle, it has the opposite effect of vasodilation. And how do we integrate this into a physiological response? Well, when a dinosaur comes along and scares the bejesus out of you, the bejeepers, sorry, then your adrenal medulla, as part of the stress response, is going to pump out epinephrine. I want to say adrenaline so badly, right? Epinephrine. And so all the blood vessels are containing plasma with a high concentration, so it diffuses out. And you want to be able to run away, so you want to increase the blood flow to the skeletal muscle. You don't want to worry about digesting anything, so you're going to decrease the blood flow to the intestines. It's obviously part of the sympathetic nervous system, but let's put it into context here while we're looking at this distinction based upon the same ligand, same target cell type, but different type of receptor on these target cells in different organs. Same tissue, different organs. Thinking of it as vascular tissue. So let's also think of another means for modulating a signal pathway. So we have receptor specificity and different isoforms of receptors. And now we think of affecting those receptors. We can either upregulate if we need to be more sensitive to any level of the chemical messenger around, or downregulate if there's too much and we need to stop reacting. So by decreasing the number of receptors, or by decreasing the binding affinity, and this is one explanation for drug tolerance. So if a person is being exposed to a drug at high amounts for a long period of time, then the cells are just going to decrease the expression. Well, first they're going to alter the proteins so that they may be bombarded by and bound by the ligand, but the protein is changed so that it has a low affinity. It doesn't like to hold the molecule there very much when it sticks. So the molecule, if you can think of this S as the binding site, it changes to an R. So now we come, and even though there's a bunch of us there, and we're all taking our turn sticking to it, we don't really stick for a long enough period of time, whereas in the higher affinity form, shaped like the S, we come and we stay there for a little period of time, and it's even with the high concentration, we're there. There's more of us there, and we stay longer when we stick to it. So you're getting a bigger response from the cell. But if there's a bunch of little arrows and they don't spend much time on the receptor, then the receptor can't get activated. So that's the affinity thing. If you decrease the binding affinity, then even though it's bumping into it, it doesn't stick properly to trigger a response. The other way is you just pull the protein out of the membrane, degrade it, and decrease the expression of the gene for it, and don't put more of the protein in the membrane, so you get the decrease in the number of receptors as part of the drug downregulation as well. So that's how people can be, one mechanism by which people can become tolerant to drugs. There are others having to do with the liver mostly, increasing the expression of the genes for the enzymes that will metabolize the drug and excrete it very rapidly. So think of termination mechanism. I've been pointing this out all along. You can't have one event to begin something in a cascade or a signaling or response biological process without thinking that there's something that's going to terminate it, right? Otherwise, the first time you're stressed, you don't turn off the stress response, you drop dead. Your heartbeat's going to just go through the roof and going to explode in your chest. So there has to be termination mechanisms. And then we want to keep in mind that diseases and drugs will modulate all of these types of biological processes we're talking about. There's capabilities across the board. I'm not saying any one disease or any one drug affects all of them, obviously. So let's look at a couple of examples of diseases and drugs that target signal transduction proteins. So we want to look at some of these the conditions that are linked to abnormal signaling mechanisms. We're looking at genetically inherited abnormal receptors in this set here, or toxins affecting signal pathways in this lower set. So in the upper portion of this table, we see the receptor, the physiological alteration, and the disease or condition that results. And then we're going to look at toxins, two of them, physiological effect rather than physiological alteration, because it's a drug or a substance effect, and then a condition that results. So remember, disease and condition, there's kind of a difference there. Conditions can be reversed or managed. Sometimes diseases can be cured or not cured, and possibly, hopefully, managed. So a receptor, for example, the vasopressin receptor, it is a membrane protein, and vasopressin is also called antidiuretic hormone. So if you can't respond to antidiuretic hormone, then you're going to produce high volumes of urine that have a lot of water in it, because antidiuretic hormone increases the resorption of water from the urine. So what happens is you have a very short half-life of the receptor. So the receptor protein may be present in the membrane, but it just doesn't stay long, so it's always less effective at triggering a signal. So the congenital diabetes insipidus, so not diabetes mellitus. Diabetes mellitus is the sugar diabetes. This is one, the insipidus is drinking a lot of fluids and excreting large volumes of fluid because you can't resorb the water in the kidney. So remember, this shortens the half-life of the receptor. So that means that at any one point in time, the concentration of receptor is rapidly being decreased. So no matter how much vasopressin or antidiuretic hormone is around, low or high amounts, you don't have the concentration of the receptor to balance it and produce an appropriate physiological response. So remember, all these events, as we've talked about, are concentration dependent, dependent upon the concentration of the ligand and the receptor. So remember, downregulation, you reduce the concentration of the receptor in the cell membrane, and you reduce the response that the cells are capable of producing, even with high levels or constant, persistent levels above physiological levels of the ligand. So a calcium sensor in the parathyroid gland, so it fails to respond to increases in plasma calcium. This is associated with familial hypercalcemia. Rhodopsin receptor in the retina of the eye, where there's an improper protein folding due to the genetic mutation or the altered DNA sequence in the gene. And this would be associated with retinitis pigmentosa. Considering toxins then, there's the Bordetella pertussis toxin and the cholera toxin. The Bordetella pertussis toxin blocks the inhibition of adenylate cyclase. So remember, you can turn on adenylate cyclase or adenylyl cyclase. Suddenly they've changed it to the 8th, but it's the same enzyme. So the adenylyl cyclase is activated, and then there's inhibitors that will inactivate it. So if you prevent the inhibition, in other words, you don't let somebody put their foot on the brake, or you cut the brake cable, right? So even though you're trying to put the brake on, you can't. And then whatever level you've got the gas pedal going, it keeps going. So in other words, the adenylyl cyclase stays active, and this produces whooping cough. So these are how these toxins were discovered, and they've been used experimentally for many years now. The cholera toxin and Botox is, I don't think that's on here, but that's another one that will inhibit contraction of muscle, so people get it injected in their face to have smooth skin on their face, but they can't move they can't make any expressions. Anyway, cholera toxin is blocks the enzyme activity of G proteins, and the cell keeps making cyclic AMP, and so ions would be secreted into the lumen of the intestine, causing massive diarrhea. So the descriptions here of these toxins are associated with the natural infection by the agents, the bacterial agents that produce these and secrete them. Now to consider pathways, we first look at an overview. So there's physiological control systems that keep regulated variables within a desired range during homeostasis. So there is some set value, like the thermostat. We always use the thermostat at our home as an example. And a thermostat is going to have some efficiency. It's not going to keep things at exactly the temperature you set it. There's going to be a range of plus or minus 1 degree or 2 degrees around that set point that you put it at, to where it's going to have its sensitivity of when to turn on and when to turn off. So this is what's meant. There is a set point, but there's a tolerance around that, plus or minus whatever, right? Because exactness is always comes with tolerances. Even for engineers that are designing pieces for our vehicles, there's always some tolerance that as the products are produced, they have to all be within certain tolerances. 1 millimeter plus or minus an angstrom or something. So anyway, there's that regulated variable that we want to keep within a desired range, and when it is in that desired range, the addition to it versus the taking away from it, the subtraction from it, there's no action required. So we have a good balance of heat coming into the system and being taken out of the system as far as staying with that thermostat idea. So there's an outside desired range and a sensor, if the regulated variable gets outside the desired range, then the sensor is activated. So somebody leaves the door open and heat gets into the house. Then the sensor is activated, turn on the air conditioner, it sends a signal to the integrating center, the air conditioning unit, so it turns on, that is going to alter the fans and the compressors such that they begin to extract heat from the air in the house, and they will influence the air in the house to bring the heat back, the regulated variable, back to within the desired range. So this control system is something that we all know as we, in this summer of 2009, it's August right now that I'm doing this original recording, we had, I don't know how many days it was, 40, 45, 50 days straight of 100 plus degree temperature. So let's think of Cannon's postulates in this context of control systems. The nervous regulation of the internal environment is key to this. There's going to be tonic control, so there's a baseline level for everything. The antagonistic control to keep it from going out of range. And then one chemical signal can have different effects in different tissues. So let's begin to think first about the tonic control of blood vessel diameter. So at any one point in time, resting or basal state, there is a certain degree of smooth muscle contraction in our blood vessel walls, different in different blood vessels throughout our body, but for that blood vessel, wherever it is, the intestine or the skeletal muscle, for example, from that prior slide, at rest, it's going to be at a certain degree of tone, of how, skeletal muscle, the smooth muscles in the walls of our blood vessels, each one doesn't just stay in a certain degree of tension. It and its neighbor are going to kind of swap off turns in keeping the tone so that one can relax and recover and then come back in to contribute. So that there isn't an exhaustion of the cellular means for contraction and contribution to tone, and to have a responsiveness if the system is challenged. So the nerves innervating the wall of the blood vessels, bringing norepinephrine to the alpha receptor or beta-2 receptor, are going to have these. Here you see the digital, right? These are action potentials so it's a square wave event. Each square wave event is going to release a certain amount of packets of the norepinephrine, and assuming there's the same concentration in each packet of same concentration of norepinephrine in each packet, with each of these digital events you contribute to the analog concentration of the norepinephrine bathing the smooth muscle cells with the adrenergic receptors on them. So these square wave digital events are converted to an analog signal coming to the receptors on the smooth muscle cells. So what can you change about this digital signal from baseline such that you can have an increased concentration of norepinephrine or a decreased concentration of norepinephrine? Remember, we're adding to this pool, and then there are nearby inactivating mechanisms, washout, diffusion into the blood and washing it away, and enzymes that inactivate it. And there's reuptake mechanisms too. So if you decrease the frequency of the digital events, decrease the frequency of firing of the neuron innervating the blood vessel, then you will effectively decrease the analog concentration of the chemical signal, the norepinephrine. So this is called frequency modulation that we're thinking about here. So at rest, there is a certain firing rate or frequency of action potentials for the neurons innervating the different sets of blood vessels throughout our body. And at that basal firing rate, there's a certain tone level of contraction of smooth muscle in the wall. And if it increases, then you increase the frequency of firing, then you increase the concentration of norepinephrine bathing the receptors in the intestinal blood vessel. This is going to bring about a vasoconstriction, a decreased blood flow. And then if you decrease the signal, decrease the concentration, you get dilation, you're going to get increased blood flow to the intestine through the alpha adrenergic receptor, to follow up on that prior example. Excuse me. Let's then think of the antagonistic control. So again, the autonomic nervous system is a prime example where you've got a different neurotransmitter in the sympathetic neuron, norepinephrine, versus the parasympathetic, acetylcholine. So they each act through different receptors and receptor mechanisms to cause opposing effects on the target cells, the target tissue, and the target organ. I wanted to go through that series of levels of structural organization because for the heart, we think about the SA nodal cells that are being targeted by these two neurotransmitters. So that's the cell. They're within the tissue, the cardiac tissue, and they're going to affect the organ, the heart, by changing heart rate. Okay, so that's the reason for doing it that way. So if we stimulate the sympathetic nerve, now remember, each of these is going to have a basal tone. So this is the gas pedal, and this is the brake. So I don't know if you've ever been in a car with somebody that have their foot on both the gas pedal and the brake at the same time. But anyway, this is the way, it's not very good on your car brakes, but it's the way the heart works. Each of these has a basal level. And for each of these, you can increase above or below its basal level, and as well change above or below basal level in the other. So you can increase sympathetic tone to the heart or decrease sympathetic tone to the heart, or you can increase parasympathetic tone or decrease parasympathetic tone to the heart. You can do each of these independently too. So if you have basal level, no change in sympathetic, but you just selectively increase the parasympathetic, then you get this effect over here. Stimulation of the parasympathetic nerves decreases heart rate. If you selectively increase the sympathetic tone, then you increase the ratio of norepinephrine to acetylcholine, and so the norepinephrine effect wins out, and you get an increase in the heart rate. Right? But if you increase both of these at the same time, so the ratio concentration of norepinephrine to acetylcholine was essentially the same, there's just a lot more of both, you still would have no effect on heart rate. So I want you to get a feel for how physiology can work, and don't get locked into these simplified illustrations. Each of these systems, portions of the autonomic nervous system, can be regulated independently. So let's go beyond this one more point to determine the heart rate from these traces. So every time we see a graphic trace, we want to look at the axes. So we have on the x-axis time in seconds. So we have an interval of intervals of 1 second, and we see 3-second trace here. And then the deflection is just the electrical charge, surface potential picked up from the skin, right? I was presuming they had the chest leads on. So it's the same in both of these, and we see that the total amount measured in each 3 seconds of the strip chart is shown. So then what we can do is count the number of individual beats. We know this is the EKG. Each one right, is a single heartbeat. So each point up here we can count. So there's 1, 2, 3, 4, 5, 6, 7, 8 within 3 seconds. So what's the heart rate? What's the heartbeat in this one? Right, 3 times 20. 8 times, excuse me, 3 times 20 is 60, so we convert seconds to minutes. And so 8 times 20 is 160. So, yeah, baseline should be about 80 beats per minute, right? And this is 120, so that's a pretty good increase. That's a selective activation of the sympathetic nervous system. Somebody just scared the bejeepers out of you. And then here you are, you just ate dinner and you're kicking back and relaxing. Actually, this is pretty slow, but here we got 1, 2, 3 times 20, 60. So that's a dramatic decrease. Now, if you're saying, "Gee, my resting heart rate is 60," that might be normal for you, or maybe not. If you work out quite a bit, pretty athletic, and you're in pretty good shape, a resting heart rate of 60 is not so bad. If it stays that low and a little bit lower, talk to your doctor about it. If your resting heart rate is too low, your heart might not be able to meet the demands. Talk to your doctor, the physician, about it. Okay, so the control pathways, comparison of local and reflex control is considered on this slide. So in the center, we have the brain that evaluates the change and initiates the response. And then we have systemic change in blood pressure that can be sensed in the carotid sinus, in the arteries of the neck, bringing blood to the brain. Brain's very selfish with blood flow, and it wants to know whether it's getting the blood flow that it needs. That's one of the reasons that this receptor is here, the carotid sinus. But there are others as well in the aorta that come straight out of the heart. But anyway, this is the one we're considering. The reflex response is initiated by cells at a distant site. So the signal goes into the nervous system and then comes out of the nervous system remote from the site of the sensor. So there are centrally located receptor systems. They're not diffusely distributed. But there can also be local changes with local responses. So if you make a skeletal muscle very metabolically active, it's going to locally vasodilate to increase the blood flow to it selectively. But this may bring about a reflex response to cause activation of the sympathetic nervous system, to cause some vasoconstriction in other areas. So, for example, a person standing on a ladder, holding their hand above their head, hammering a nail, there will be local vasodilation to increase blood flow in that extremity, the muscles being activated, but then the central triggering response activating the sympathetic nervous system can raise blood pressure. So somebody that already has high blood pressure and doesn't have a really good vascular system can be placing themselves in a very vulnerable situation for a cardiovascular event simply by climbing up a ladder and hammering a nail into the ceiling. So I want to put it in perspective here for these control pathways and how it can relate to pathological conditions. So the reflex control is one that we can consider in this slide. So there are steps in a reflex control pathway: stimulus, sensor or receptor, afferent pathway, integrating center, efferent pathway, target or effector, and the response. So there is a response loop, and then there is a feedback loop. So let's think of these control pathways, starting with the receptor. There are multiple meanings of this word receptor. There's the membrane protein that we talked about, or the cytoplasmic protein, or the nuclear protein, or specialized cells or structures that convert various stimuli into electrical signals. So there's sensory proteins, there are receptor proteins, There are receptor cells, there's receptor tissue, receptor organs, right? So if we this isn't such a problem if we remind ourselves of the levels of structural organization. In each of these cases, whatever is being sensed by the receptor is ultimately being accomplished by at the molecular level, the chemical level. There's some chemical that is capable of being altered by the stimulus, and then that chemical alteration brought about by the stimulus is somehow transduced into a signal at a grander and grander scale. So the central receptors are in the, or close to the brain, such as the eyes for vision, ears, nose for smell, tongue for taste. And then there's peripheral receptors that lie outside the brain. So the term that I was using is centralized. Central is really more centralized. It's centralized to a single restricted location, not diffusely distributed in all the blood vessels. When I said central here, I didn't mean central as far as being within the nervous system, as they're defining here to distinguish central receptors close to the brain, although those blood vessels, that blood vessel still is closer to the brain than this one, right? Anyway. And then the peripherals, so that lie more remote or distant from the brain, but definitely outside of the nervous system. So there's chemoreceptors, the pH, the gases and chemicals, or osmoreceptors for osmolarity, thermoreceptors for temperature. So you see that these first three have both central and peripheral distributions. And then there's baroreceptors for blood pressure. That's what we saw in the carotid sinus. As the carotid sinus stretches, or the aortic stretches, because there's higher pressure, then the baroreceptor nerve endings inside the blood vessel wall are going to be stretched more and send a stronger signal, higher frequency of firing rate into the brain. Proprioception for body position, and then mechanoreceptors for pain, vibration, and touch. So obviously, when we discuss, consider, and study the sensory nervous system in detail, we'll look at these much more closely. Well, now let's think of that response loop, and what we're going to think about, or use as an example, it's a thermostat, just like we talked about, but this one is for controlling water temperature. So a non-biological response loop is a good way to go. So there's all these parameters, and the reflex pathway is the what we'll be considering in parallel with this, and the series of slides coming up adds these one by one. So first, in the reflex steps, the water temperature is below the set point. For some reason, heat is leaving the system, and the desired temperature is not sustained without some intervention. So we have water temperature, it's 25 degrees C, and the stimulus that comes into this system is that heat is removed from the water by whatever means. So we have a sensor or receptor. The thermometer senses the temperature change and a decrease in this case. And then we have the afferent pathway bringing the affect. So the sensor picks up the stimulus, and that affects the system. So the affect is going to be in the afferent conducting aspect of this system. So the signal passes from sensor to control box through the wire. Then there is the control box itself, such as might be part of the brain in the biological system that we considered previously. The control box is programmed to respond to temperature below 29 degrees. So this is the integrating center that we would think about in a biological system. Could be neurons of the brainstem for blood pressure control. The signal passes through wire to the heater. So there's an efferent pathway. We're going to produce an effect in response to the affect, right? So the affect of losing heat will effect or bring about the generation or the addition of heat to the system. The heater turns on, and you get a target for the efferent pathway or an effector. This is the heating element is the effector to effect heat addition back into the system. And so as this is allowed to be ongoing, it should sustain within a tolerance, plus or minus a degree. So at 29, plus or minus a degree, whatever it would be. And the water temperature is going to increase back to the set point, 29, from 28 to 30 degrees, right? And begin the feedback to turn off the system. So before getting to the feedback part, let's think about set points. So there's a tolerance, meaning there's an oscillation around a set point. So if you were to measure a parameter over a period of time, collect a sample data set, the larger the data set and the more numbers, the more frequency, the smaller the frequency interval. If you measure it microseconds rather than hours, you're going to get more numbers over an hour and have a better representation of all the data points over that period of time. And then if you get the average, then you would think that that is the set point of the function, correct? So the mean value of a large data set over a period of time would be considered as the set point. And then the highs and lows would be considered as the tolerance or the normal range of the function. So the maximum highs and minimum lows would be the range of the set point. So, the range of the function, the tolerance around the set point. And so the response loop is going to turn on in between these highest and lowest levels to bring the system back within the normal range of function. Let's then consider feedback loops. So the negative and positive feedback are the two classics. There's a feed-forward control that refers to anticipatory responses. And then the response counteracts the stimulus, shutting off the response loop in the case of the negative feedback. So the initial stimulus causes a response, and then you're going to have a decrease in the stimulus, and the response loop then will shut off. Continuing with the feedback loops, the response reinforces the stimulus. This would be the positive feedback, correct? Negative feedback was on the prior slide, where the effect leads to a shutdown of the system close to set point. Now in positive feedback, the effect is going to drive the system to greater levels. Typically it's an increase, enhancement. So it sends the variable farther from the set point. So you have the initial stimulus, a response that ends up enhancing the stimulus such that there's a greater response, and then there's some outside factors required to shut off the feedback cycle. So there has to be some other biological process, biophysiological parameter that is going to shut this system off. You can't have a system go uncontrolled. So the classic example of a positive feedback is labor and delivery. So there is a cervical stretch as the baby drops lower in the uterus to initiate labor, and this sends a sensory signal up to the nervous system, to the hypothalamus, to release oxytocin from the posterior pituitary gland, and that is distributed through the blood and has target cells in the myometrium, the muscular lining of the uterus, and it causes uterine contractions until ultimately, of course, you have the delivery of the baby. And so that external factor, the emptying of the uterus, actually, the collapse of the uterus wall, and the termination of the stretch of the cervix will turn off the system. But up until the point that you have the delivery of the baby, this positive feedback system continues. So you get the initial stretch, oxytocin release, uterine contraction, push the baby against the cervix to stretch it more, and then you get more cervical stretch, more oxytocin, and so on and so forth. And then once you get the external event, the delivery, and the recoil of the cervix, the loss of the cervical stretch, then you get finally the shutdown of the system, so the oxytocin is not being released at such a high level anymore. It continues for a while to contract the uterus and stop the bleeding. There are set points. So some of you might have heard of Pitocin, which is a synthetic form of oxytocin that's sometimes given to induce labor, sometimes given after delivery to enhance the uterine contractions to stop the loss of blood through the uterus. You don't want it to become a hemorrhage. Continuing then with this idea of set points and tolerance around that, there can be even larger variations over larger scale time periods called circadian rhythms. So there's shorter term ones. I think on the prior scale, didn't we see time? So if this was in minutes or hours, and we're looking at a circadian rhythm across the day, the daily rhythms, these would be called ultradian rhythms. So nothing is ever at one single point. It's always down and up around the set point. So this would be an ultradian rhythm of a biological measurement. And then over a period of a day, midnight, noon, and midnight, you would have the daily cycle. So this would not be a perfectly smooth line if you were able to represent the shorter time interval measurements. This is a wavy line because, say, it's oral body temperature. So it's changing. It's not exactly at the exact same measurement at every point, every minute, right? But it's always going to be around on either side, above or below, some average for that period of time. So there are these different contributing regulatory mechanisms for the ultradian, but then some harmonic, if you will, on top of the ultradian to produce a daily rhythm. And then, of course, there can be, like the menstrual cycle, that are monthly rhythms, annual rhythms, and things like that. So in other words, biology is not constant. Continuing with this idea of set points and rhythms, we can use plasma cortisol as an example. Notice the units of plasma cortisol are micrograms per deciliter. Again, each time x-axis and y-axis we want to take a look at. Sorry about that. So it's time, and this is 12-hour intervals of time on the x-axis here, and oral body temperature in degrees centigrade. So 37 degrees centigrade is considered to be normal body temperature. And in this set of individuals, or particular individuals, 37 did not turn out to be the average. It's more like a 36.8 8 or something. So we have same 12-hour time intervals on the x-axis for this graph, and the plasma cortisol in units of micrograms. That's a U, but it should be mu for micrograms per deciliter of blood. So can you convert this to milliliters? Can you convert it to centiliters? So make sure you can easily work with these to convert them. Because obviously there will be questions that will require you to recognize that that is what is different between two different sets of data points, and can help to explain why you're seeing a difference. So here we are seeing a range of plasma cortisol basically from roughly 5 to just below 20 micrograms per deciliter. So from midnight to 9 a.m. We see the range of the lowest to the highest value of plasma cortisol. And then this dips to just after lunch basically, and then comes back up again a bit just before dinner time or shortly after dinner time, and then it dips over the nighttime period back down to the low level, and the lowest being roughly around 4 or 5, 3 to 5 o'clock, I guess, whatever time of day would be. Well, that would be 6 hours there, wouldn't it? Yeah, so about 3 o'clock. About 3 a.m. you would expect to be the lowest level. So this means that you don't always have the peaks and troughs at exactly the same time for each biological rhythm. Here you can see peak temperature just before dark and trough temperature just before daylight, and the plasma cortisol is shifted by about a 3-hour time frame from the temperature. So they're not all synchronized, right? Each has its own unique regulatory mechanisms. Now let's think about speed and specificity. So there's a comparison of neural and endocrine control on these two concepts of speed and specificity for regulatory mechanisms. So let's take a look at the property in the left column, and then the neural reflex in the center column, and the endocrine reflex on the right. So for specificity as a property, for the neural reflex, each neuron terminates on a single target cell or on a limited number of adjacent target cells. The endocrine reflex, most cells of the body are exposed to a hormone. Of course, the response depends on which of those actually express the receptor on the surface. And then the nature of the signal. For the neural reflex, the electrical signal passes through the neuron. Then chemical transmitters pass the signal from cell to cell, that digital to analog converter. In a few cases, signals pass cell to cell through gap junctions. The endocrine reflex, the chemical signals are secreted into the blood, into the interstitial fluid, and it diffuses into the blood for distribution throughout the body. As far as speed, the neural reflex is really very rapid. The endocrine is going to be dependent upon the distribution of the signal and the onset of the action based upon spatiotemporal features of the localization of the cell of origin and the target cell. So typically slower than neural. The duration of action is usually very short for the neural system. The responses of a longer duration are mediated by neuromodulators. So the way that we want to think about this is that the neural reflexes will be primarily responsible for the very fast and short-acting types of responses. Still capable of lingering responses, but responsible for getting that initial response generated. Right? If you got to turn and run and get the hell out of there, then you want your nervous system to do that for you, right? You don't want to wait for a hormone to get released and distributed through the blood. Comes from the adrenal gland and then has to go through all the venous system to the heart, and then through the lungs, and then back to the heart, and then finally out through the aorta, and then distributed. And that's still pretty fast, but it's a long distance, to get distributed to the final target sites. So it's coordinated. The neural gets the quick, short-lived, with some duration, the intermediate and long-term duration, and then the endocrine reflex picks up with the intermediate and definitely sustained responses. The coding for the stimulus intensity, each signal is identical in strength. That's the digital signal, the action potential, right? The stimulus intensity is correlated with increased frequency of signaling. So the frequency modulation of the digital signal is the coding that we get. It's more complicated than that, but the frequency modulation, is different. And then the stimulus intensity is correlated with the amount of the hormone secreted. So do you remember the radios being in FM and AM? We still have them that way, right? In our cars, we have FM and AM choices. So frequency modulation with no change in amplitude. And then amplitude modulation without a change in frequency. So you can get a stimulus intensity correlation with the amount of hormones secreted in an analog system of amplitude modification, AM type of modulation, and the digital is affected mostly by frequency. Let's review the control pathways then. A simple endocrine reflex, the internal or external change. The endocrine system sensor integrating center is associated with that, and then we get the efferent signal, the hormone, to the effectors for a response. The simple neural reflex, there's again the internal or external change, a receptor, the afferent path, the sensory neuron, bringing the affect to the neural system integrating center. In contrast to the endocrine cellular sensory integrating system there. Then the efferent neuron will send the action potentials to the effector directly to get the response. And then a complex neuroendocrine reflex that will have a neural component, then the hormonal or endocrine component. We see the internal external change, a receptor, the afferent sensory neuron, the neural integrating system, the efferent neuron or neurohormone that then will be distributed through body tissues. The endocrine integrating center will be the origin of the hormone, the efferent signal number 2, being distributed through the body tissue fluids to the effectors and then to the response. Let's review then some of the control pathways. They get ever more so complicated as we go through this series. So we'll look at each of these separately. So some basic patterns of neural endocrine, and neuroendocrine control pathways. There I go, exaggerating. Anyway, so the first one, control pathway, is the simple neural reflex with a stimulus, a receptor on the afferent neuron to transduce that stimulus into a codified signal, the action potentials, frequency of which can be modulated, so it's a digital signal, and then the analog signal, the chemical release and local concentration on the target cell within the CNS, the integrator, where the efferent neuron will release, again, you'll have digital to analog conversion there, and the target cell producing the response. So the simple neural reflex is neuron 1 and neuron 2, the afferent and efferent neuron. A neurohormone reflex, we still have neuron 1 and neuron 2 with a neurotransmitter in between, but neuron 2 is going to have a hormone be released, and that will be washed away by the blood and delivered to the target cell to bring about the response. The neuroendocrine reflexes, first of which, the number 3 in the series here, but the first of the neuroendocrine reflexes, again, the stimulus with the receptor on neuron 1, neurotransmitter between neuron 1 and neuron 2, and then a transmitter between neuron 2 and a target effector cell that will send off signal 2, hormone 2, to be distributed systemically through the tissue body fluid, or the interstitial fluid in the plasma, to the target cell to get the response. And then again, we have the stimulus with receptor on neuron 1, transmitter to neuron 2, neurohormone to a second hormonal cell. In this case, it's an endocrine cell rather than a neuroendocrine cell. And it will release to the interstitial fluid, and then the body fluid tissue distributing it to the target cells to bring about the response. So note here we now have 2 vascular systems in this neuroendocrine reflex, vascular system 1 and 2. This is the systemic, and this will be typically a local one. And with the endocrinology, we'll talk about that, of course, in more detail. Number 5, the stimulus. Again, neuron 1 with neurotransmitter between neuron 1 and neuron 2. The afferent and efferent neurons still. And neurohormone, vascular bed 1, endocrine cell 1, and then hormone 2, and then vascular bed 2, endocrine cell 2, hormone, well, they're calling it hormone 2, but this is hormone 1, hormone 2, and hormone 3 in my book, right? So please, in your mind, change that to a 3. We'll call this a 3-cell, 3-hormone system when we talk about the neuroendocrinology. And then the final vascular delivery system to the target cell for the response. And then a simple endocrine response is the stimulus directly on some type of a receptor on the endocrine cell itself, regulating the synthesis and secretion of the hormone, and then the body fluid tissues responsible for distribution to the target cells to bring about the response. To review then, comparison of the neuroendocrine, and the endocrine reflexes. So a sensor or receptor for the neural is a special and somatic sensory receptors. For the neuroendocrine, there's special and somatic receptors, sensory receptors as well. For the endocrine, it's the endocrine cell directly. The afferent pathway in the neural and neuroendocrine are the afferent sensory neurons. There is none in the endocrine because it's directly on the endocrine cell. Integrating center is present in all 3 cases. It's the brain or spinal cord for the neural and neuroendocrine, but the endocrine cell directly for the endocrine reflex. The efferent pathway is going to be an efferent neuron, so the electrical signal, the digital signal being converted to the analog signal, the neurotransmitter, and the that has been released. Then the neuroendocrine has the same, whereas the endocrine is the hormone synthesized and secreted in response directly from the stimulus. The effectors will, for the neural, is typically muscles and glands, sometimes the adipose tissue. The neuroendocrine is, you just have to think of most cells of the body. We get into more details, so there's some selectivity, but in general, the entire neuroendocrine system is going to have effects on most of the cell bodies. The endocrine as well in most cells of the body. And then the response for the neural, you're going to have muscular contraction, secretion So those are the two primary ones. And then maybe some metabolic effects. For the neuroendocrine, you're going to have enzymatic reactions, membrane transport, and cell proteins that will be affected. And that's, of course, same as the endocrine because we're talking about the final part, the endocrine and endocrine in each of these. So then what we have considered in this set of 2 lectures, cell-to-cell communication, considering the electrical signals, chemical signals, and 4 methods of cell-to-cell communication. We talked about the signal pathways for signal transduction, amplification, second messenger systems, different receptor enzymes, G proteins, very important, integrins, and then the ligand-gated ion channels. And then novel signal molecules in this second presentation, calcium, nitrous oxide, carbon monoxide, hydrogen sulfide, and some of the lipids. And modulation of signal pathways, we saw the agonist versus the antagonist, upregulation versus downregulation. Then we went on to the control pathways, talked about Cannon's postulates. You might need to read the text to get more details on exactly who Cannon was and how it's relevant, but that's fine. Local control, reflex control, and then the feedback loops, the negative feedback, positive feedback, the feed-forward control, and circadian